Hey, welcome back, everybody. We're going to jump into our next set of notes. So, without further ado, I'm just going to shrink myself down and we're going to jump right into it, okay? So, our next set of notes, we're actually starting a new module. So, we're now talking about life's matter and energy. So, as we go through this, I do want to give you a quick warning, short little warning. This is the part of the class that most biology students, in my experience, have struggled with. It's very abstract. We're looking at organic chem and atoms and molecules, and it's kind of hard to envision some of this. So be aware of that. Make sure you use the office hours as much as possible because when you get confused on this, it'll kind of be a hindrance for the next couple units, and you're going to want to get a good firm understanding of what's going on. So if you don't understand something or if you're not sure about something, come to office hours and let's make sure that you do. Okay, so this is module 3, lesson 1. You can see the learning objectives off to the left. They're also on Canvas. I'll let you look through those on your own. Our focus today is going to be photosynthesis, an overview, and we're going to do this pretty quick, but keep in mind, make sure you use those office hours and come back. The good news about this is they recently changed the curriculum, so you don't have to remember as many details. There's all these enzymes and stuff that are involved with these processes. And in the past, you had to kind of remember some of the key players of the enzymes. Now I think they only make you memorize maybe one or two or be familiar with one or two. And their focus is more on the overall picture of how this works. Okay, so that's the good news. The bad news is, even though they made that change, it's still a struggle with some students. Now, some students do great at this because they really like this type of stuff, but others tend to have a few issues with it or they don't understand things. So make sure you come and see me if you're having problems. All right, let's go ahead and get started. Just as an introduction, this kind of all relates, right? So we'll start exploring how these nutrients are made by plants. So in this lesson, we're going to explore how nutrients and molecules kind of cycle through an ecosystem, but we're also going to look at how it starts, right? Plants are the producers. We learned that in the last module. And they take the sun's energy and they're able to convert the sun's energy into chemical energy found in things like glucose. So we'll kind of look and we're going to see how some of this relates to things like the carbon cycle and even the water cycle and some of those different cycles. So when we're doing this, an example would be the carbon cycle. As I just mentioned, carbon is being taken in by the plant. The plant then uses the sun's energy and it produces sugars, which are used to make structures inside the plant like cellulose and starch and glucose for energy. And then it releases oxygen in the process. So this kind of all ties together, even with the previous unit, even though they look like they're separate units and separate topics and separate, subjects, they actually kind of all have some relation with one another. And you're going to see a lot of tie-ins with this, okay? All right. So just as a review, chemical reactions are happening inside your body right now. They're anabolic and catabolic reactions, which means they're building things and breaking things down. So they're responsible for breaking down substances into smaller pieces, like through the digestive process. And then other chemical reactions are using smaller substances to build larger ones, like your muscles in your body and so on. In every chemical reaction, there's products and reactants. The reactants are the substances that are initially present and are used to make the products. Therefore, the products are the substances that are created in the chemical reaction. So we're going to see this right away. I think in one of the very first slides that we're going to look at, you're going to see the balanced equation for photosynthesis. So we'll kind of look at that, but keep in mind this is a multi-step, many enzymes involved, but we're just going to kind of look at the overview of it. We want to look at the big picture of this, and if you want to get into that, then take some organic chem in college or something like that, and you'll learn a lot of the details, okay? So as an ecological review, as I mentioned earlier, producers are the organisms that are able to create their own energy and provide the foundation of energy for entire ecosystems. The most common producer are plants, and since we're talking about photosynthesis, we're going to be focusing specifically on plants. They harness the energy from the sun and make chemical energy in the form of something called glucose, and then They basically can store those in the form of starch, and then we can use them later on. They can also convert them into cellulose that makes up the cell walls of the plant. They get their energy they need by, other organisms even get their energy from the sun ultimately because they end up eating the plants or they eat an animal that ate the plants. So this is kind of passed down through these food chains and food webs. And ultimately, the producers are responsible for supplying all the energy needed for a particular ecosystem. All right, so now let's jump into the nuts and bolts. So the chemical reaction for photosynthesis is 6 molecules. This is the number of molecules, and this is the molecule here of H2O. That's water. So 6 molecules of water reacts with 6 molecules of carbon dioxide. That's CO2. And that produces 1 molecule, this is one big molecule, of C6H12O6, which is glucose or a sugar. And then it also produces as a byproduct 6 molecules of O2, which is oxygen. So that's the equation. The arrow shows the direction that the reaction is occurring. So these are the reactants, and this is what's being produced over on this side. So 2 reactants, 2 products. So the arrow represents the reaction taking place. It separates the reactants and the products in the chemical reaction. Photosynthesis is powered by the sun, and sometimes they would even draw another arrow kind of coming in from the sun, showing that sun's driving this reaction. And it happens, this photosynthesis is going to occur when sunlight is striking a pigment called chlorophyll, which is found in the chloroplast of cells. It says one is pictured to the left, but it's actually on the next slide because we couldn't fit it on here. So let's take a look at one of these chloroplasts. So chloroplast is right here, and I'm going to be referring to this on the next couple slides, just kind of pointing to some different parts of it. And there's a little close-up view of one of these thylakoids here. So photosynthesis can be divided into 2 stages. You have the first stage, which is the light reactions or the light-dependent reactions. And then the second stage, which is usually called the Calvin cycle or the light-independent reactions. You might also hear it called dark reactions. I don't really like that term because it sounds like it has to happen in the dark, and that's not true. It just doesn't require light to occur. Okay, so let's go ahead and we'll start looking at these one at a time. Photosynthesis is a 2-stage process. The first stage, known as the light-dependent reaction, uses light, and it energizes a carrier molecule and basically helps us produce something called ATP and NADPH. And it does this by splitting water. The splitting of water kind of replaces the electrons. We'll get into the details a little bit in the next couple slides. The ATP and NADPH electron energy carriers then generated in this first stage are transferred to the next stage where the Calvin cycle occurs. So the chemical reactions of the light-dependent process occur in the grana of the chloroplast, and they are in these stacks of thylakoids, and they basically use light to kind of raise this up. So let me kind of point some of this out, and we'll do it on the next page as well. So this first stage, right, the first stage is happening in these granum. These granum are these stacks of thylakoids. Here's one thylakoid right here, and if you look at these little red dots that are down here, those are enzymes, and each of them have a funky name. The only one that you probably should be aware of is this last one, which is called ATP synthase, and it's called that because it synthesizes ATP. But all these enzymes along here, these proteins that are embedded in this membrane, help produce ATP. Okay, it's called the electron transport chain. And that's found in these grana. Now, the Calvin cycle is going to happen over here in the stroma. Outside these thylakoids, it's just this fluid matrix in which other enzymes are kind of floating around in that help do the Calvin cycle. So we're going to look at these kind of one at a time. I'll go into a little bit more detail, and I'll be referring to the chloroplast picture as we go through. So the light-independent process, also known as the dark reactions, I don't like that term, or the Calvin cycle, are basically going to occur when ATP and NADPH are then transported from where it's made over here and inside these granum out into the stroma. Okay, so those enter the stroma, and then there's a whole bunch of enzymes inside the stroma that pick up those molecules and basically do a further step during the Calvin cycle to start to string together carbon from carbon dioxide into glucose or sugars. So when these are basically transported over, they can use that energy to kind of start forming covalent bonds between carbons to form carbohydrates or sugars. The carbon used to make these carbohydrates comes from carbon dioxide, which is obtained from the environment. The reactions of the light-independent process take place in the stroma, as I mentioned. And then the stroma is basically this colorless fluid surrounding the granule and the chloroplast. And again, it's kind of this stuff floating around out here. It's labeled right there, stroma. And then this is where light reactions occur, and this is where the Calvin cycles occur. All right, so let's look at the light-dependent reactions really quick. We'll go through this and go into some of the details. And remember, we're trying to keep this an overview. So again, if you get confused, come and see me. So what are the light-dependent reactions? So what's going on during this? Now, this is kind of a cartoonish drawing of a chloroplast, and it just splits it down the middle. This isn't really how it's organized. And you got these thylakoids. That's where the light-dependent, so this is the light reaction part, and this is going to be the light-independent reaction part, or the Calvin cycle. So we'll refer to this and kind of look back and forth. But keep in mind, it's not set up exactly this way. It's kind of a cartoonish way of looking at it just to help us understand. So there's lots of complex steps and processes that are occurring during the two parts of photosynthesis. So we're going to look at a quick summary and pay attention to what the plant needs or what it's bringing in to cause the reactions to occur, and then what's being produced. So the first thing that's happening is we have light, right? By absorbing light, pigments called chlorophyll, these pigment molecules are acquiring some of the energy that is carried by the light. And when this acquired energy strikes the chloroplast, it's going to basically excite these electrons to a higher energy level. Those electrons, when they get excited, are then going to be passed to something called an electron acceptor, which then donates the electron to the electron transport chain, which is where ATP is made. So light's coming in, right? Light's coming in, and we're able to produce some ATP in this process. And then we're also able to take some NADPs and produce some NADPH, which is basically a carrier. It's carrying the hydrogen ion, which sets up a hydrogen ion concentration gradient to generate ATP. And then some of that can also be used in the Calvin cycle as well. So that's kind of what's going on here. So we got an influx of light, and we're making some ATP and NADPH. So what is this right here? Why do we need water? Well, let's look at that. The electrons, once they're excited and passed off to the electron acceptor, have to be replaced. So electrons that move from the chlorophyll molecule to the electron acceptors must be replaced. The replacement electrons are provided by the splitting of water. So we take water in, H2O, and it's split into hydrogens and oxygen. And oxygen is one of the byproducts. So the water molecules split to replace the electrons, and oxygen is produced. And after the oxygen is made, it diffuses out of the chloroplast and leaves the plant and goes into the atmosphere. So that's the byproduct that's made. So if I back up, this water comes in, it replaces the electrons right here, and then we just have the oxygen left, and the oxygen is released as a byproduct. All right, moving on. Making ATP in the light reactions, an important part of the light reaction is the synthesis of ATP. During chemiosmosis, the movement of protons through ATP synthase into the stroma release the energy. So that's where the one enzyme that's mentioned is ATP synthase, and that's where ATP is produced. Now, what is ATP? ATP is adenosine triphosphate. Think of this molecule as an energy molecule. And if you think of it, sometimes they call it an energy currency. If you look down here, ATP is an important molecule in living things because it's the energy currency of the cell. So if you think of this as currency, another word for currency is money. So one way I think of it is ATP are the lower denominations of currency. It's a dollar bill. And think of this as an exchange system where there's no change. You can't get any change back. So if something costs, 50 cents, you have to use a dollar, you give them a dollar, and they get to keep the change. Well, that would be terrible if we had a $100 bill, right? If we had a $100 bill, and something costs 50 cents and I gave them a $100 bill, they're going to keep the 100 and I'm going to get a 50-cent product. That's not fair. So ATP is a smaller denomination that we can use. And I say this because what we're making right here is ATP, but we're going to send that ATP to Calvin cycle and we're going to change it in to a higher denomination bill. That higher denomination bill is going to be glucose. Okay, so glucose it takes a lot of ATP to make glucose, but it's worth a lot of ATP as well, and that can come in handy when we do cellular respiration, something we will be covering later in the year. So it's a great short-term energy storage molecule, and it's relatively unstable. It wants to donate these extra phosphates that it has, and the energy can be released when it loses one of the phosphate groups, and it can be used to do different things. It can be used to cause a muscle contraction inside a cell. It can be used to move something around inside the cell, and so on. So let's now move on to the light-independent reactions. Living systems cannot directly use light energy, right? It has to be converted to a type of chemical energy. And we just mentioned that we're producing ATP and NADPH. We can then use the NADPH and ATP in the Calvin cycle. Now, you don't have a bunch of enzymes standing in a circle doing this. It's just kind of drawn as a cycle to help us understand it. So all these enzymes are going to be floating around in that stroma, right? And they're going to be doing these various procedures. It's a multi-step process to do this. And we're going to keep it super simple and super basic. So we're going to use the ATP and NADPH to form carbon-carbon bonds in simple carbohydrates like glucose, different sugars. They're basically just a simple monosaccharide sugar. This stored energy can then be released on demand in other metabolic processes, i.e., we're talking about cellular respiration, and we'll talk about that later, as I mentioned. So the glucose we're making is basically a bunch of ATP. So if I take a $21 bills and give it to somebody and they give me a 20 back, that's what we're basically doing. We're using this ATP to make a larger denomination bill that we can store, that we can put away. It's easier to carry around, 5 20s than it is 100 singles. Okay, so we're kind of making these storage molecules where we can store this energy and then break it down and turn it back into ATP later. Okay, so the reactions that ATP and NADPH use to convert carbon dioxide into carbohydrates makes up the second stage of photosynthesis, and it's known as the Calvin cycle. So these reactions are known as the dark reactions. Again, not really a fan of that term. Or the light-independent reactions. That's a better term. Or the Calvin cycle. The ATP and NADPH produced in the light reactions drive the second stage of photosynthesis, the Calvin cycle. And in the Calvin cycle, carbon dioxide is brought in, and it's basically added to a 5-carbon molecule, and then it gets recycled and turned into a 3-carbon molecule, and then every once in a while, 3-carbon molecules pulled out. When we get 2 of those pulled out, we can formulate a 6-carbon sugar, and then the rest of it gets cycled back around, which is why it's called a cycle. Now, one misconception is that these reactions occur only in the dark. This is not true. That's why I don't really like that term. It's just that they don't require light. They just require the products from the light reactions to do. So if the plant's not exposed to light, then it's not going to make ATP, and it's not going to make NADPH, and it will shut this down. But if this happened and we have a store of ATP and NADPH, this can happen with no light going on. Okay, it just needs the products from this. But if there's no light for a long time and we run out of ATP and NADPH, there's a problem. All right. So this process in which we're adding carbons to these 3-carbon sugars to make a 6-carbon sugar eventually called glucose is called carbon fixation. So the Calvin cycle, which occurs in the stroma of the chloroplast, is a series of enzyme-assisted reactions, and you don't have to know all these enzymes. This is that part that I said you used to have to remember, but now we don't need to. It's basically enzyme-assisted chemical reactions that produce a 3-carbon sugar. Most of the 3-carbon sugars generated in the Calvin cycle are then converted to a 5-carbon sugar to keep the Calvin cycle operating. But every once in a while, they pull out some of these 3-carbon sugars, and they're used to make a 6-carbon molecule called glucose, which is basically energy stored for a later use. Again, think of it as a higher denomination, right? A $100 bill instead of a whole bunch of ATPs, we can just get 1 glucose. So the carbon cycle connection, what's the connection here? The carbon fixation transfers carbon dioxide from the atmosphere into organic matter. It remains in biomass, coal, oil, gas, plant remains until decay or digestion, or burning it releases it back into the air. So this is kind of linking photosynthesis to what we just previously studied in Module 1 and 2. So as the amount of plant growth changes, so does the atmospheric carbon dioxide levels. Now, there's several key environmental factors that can affect the rate of photosynthesis, and this is going to help you with some of the assignments in Canvas. Light intensity can affect it. Carbon dioxide levels can affect it. Temperature, the amount of water available can affect it. So let's look at a few of these real quick, and we'll look and compare it to a couple of graphs. Okay. So I'm gonna move myself over here real quick so you can see the graph a little bit better. So if you take a look, the rate of photosynthesis increases as light intensity increases because more electrons are excited in both photosystems. However, at some point, all of the available electrons can get excited. Now, we're looking just at light intensity here. This is that first graph. And if they're all being used, right, if they're saturated, if light intensity can't make the electrons that are getting excited work at any faster rate, then you'll notice that the graph kind of flattens out. So as the rate of as light intensity increases, the rate of photosynthesis is going to increase. But once all the available electrons are excited, and there are no more to excite, no matter how much more bright the light or intense the light could get, you'll see it starts to flatten out, okay? The same happens with carbon dioxide. So if we take a look at carbon dioxide, just like we talked about, increasing amounts of carbon dioxide will cause the rate of photosynthesis to increase, but eventually, once all the enzymes are working to do the carbon fixation and all the carbon dioxide is being used, and we're at maximum saturation and all the enzymes are doing the work as fast as they can, you see it'll start to flatten out. So what's going on here with temperature? Notice it goes up, and then at a certain point, it actually starts to drop. Okay, so let's take a look at temperature real fast. As temperature increases, the rate of photosynthesis will increase to a maximum, and then all of a sudden you see it start to drop off. With a further rise in temperature, you see it start to kind of drop down. This is because many of the enzymes that catalyze a reaction become ineffective. And what happens is these enzymes are proteins. So proteins of a particular shape, right? If it gets too hot, the protein changes its shape, and once it changes its shape, it kind of stops functioning. So it's called the denaturization of proteins. So if these enzymes become denatured or their shape changes, they no longer function properly. And if they stop functioning properly, photosynthesis starts to drop off down here, right? And we start to see this fall off. So that's why temperature looks a little bit different. All right. Let's move on. We're going to look at the last slide, which is going to focus on your assignments and stuff like that. So if you remember, just like in all the modules, make sure you do all the self-checks. Those are like little mini quizzes. Remember, you can do those multiple times. If you get it wrong the first time, you can take it again and again, and it will increase your grade until you have 100%. Make sure you watch all the videos. That'll help you, especially with the 1.5 assignment, the photosynthesis science notebook. You'll have to watch the videos and then respond to a writing prompt. There's also a photosynthesis review. It's a little bit of a long review sheet, but it should help you understand photosynthesis a lot. So make sure you look at that. And then one of the things we're going to look at probably on Friday is we're going to look at this lab demo, this leaf disc lab. And I wrote Friday on there because we're going to kind of go over it on Friday. It's basically a video from Mr. Anderson of Bozeman Biology. I use his videos a lot. And he kind of shows a leaf disc demo, and it's a little 5 or 6 minute video. And then I give you some mock data all filled out in a data table, and you just have to answer some questions about that data. So take a look at that. We'll go over that a little bit on Friday as well. And then there's another discussion board that you can add to the photosynthesis. You guys usually do pretty well at that. Pretty easy assignment. So not too difficult of a week. And we will be going over the leaf disc lab a little bit on Friday, so you should have a pretty easy week. And remember, come and use those office hours if any of this gets confusing, so I can help straighten you out and make sure that you guys are doing things the right way. All right, guys, have a good day.